Hi, everyone. Welcome to the Voice of Pancreatic Cancer podcast. I'm your host, Miranda Weinberg, and I'm joined today by Dr. Susan Tsai. She's co-hosting with me. If you have listened to the episode before, you probably recognize her. She's joined us a lot. If you don't know about the St. Magwitz Foundation, we are a nonprofit that's committed to the awareness, prevention, and cure of pancreatic cancer. And we work very closely with the Medical College of Wisconsin, one of our partners. Dr. Tsai and I are really excited today to welcome Dr. Nikki Lytle. She is an assistant professor at the Department of Surgery at the Medical College of Wisconsin. Welcome, Dr. Lytle.
Thank you so much for having me today, Miranda.
Okay, so Nikki, one of the things that I think maybe the listeners do or do not know is I often get this question in clinic: how do cancers develop? What could I have done to stop a cancer, et cetera? And for pancreas cancer, I usually tell people, there's some known risk factors that are associated with pancreas cancer. Smoking certainly is one of them, family history and other things. But a lot of the times we don't know exactly what's triggering the cancer, what's really causing the cancer. And in general, I think, when we think of the cancer itself, I think there's kind of two different pathways that take us there. Either they happen because there is a cyst in the pancreas that turned into a cancer. And I often tell people that a cyst to the pancreas is like having a polyp in your colon. It's like a pre-malignant growth that we just have to watch. And sometimes we don't even, the cancers arise separate from the cystic disease. And we spend so much time focusing on the cancer itself. But there's this whole nother side of pancreas cancer biology that I'm not sure the listeners know about, which is the tumor microenvironment. And so, can you kind of introduce that to our listeners and just kind of tell us what that is and why specifically in pancreas cancer it's important?
Absolutely. It's there's been a lot of sequencing data recently in non-tumor bearing people. So these people, maybe they pass away sporadically in a car accident and what's interesting is we now know that people by the age of 30 have a lot of cells in their body that have the mutations that we know drive cancer, but those cells are just sitting there. They haven't quite become cancer yet. And as we age, we accumulate more and more of these cells that have these mutations, but again, they're not driving cancer, they're just sitting there. And so the question is what then makes those cells either decide to stay as these pre-malignant, non-aggressive cells or trigger them to flip into a cancerous phenotype? And so, a lot of my interest is trying to understand the heterogeneity or the phenotypic differences between these cells that will push one into a malignant aggressive state. And one of the contributing factors that we believe drives that is called the tumor microenvironment. And the tumor microenvironment, it largely consists of cells within our body that aren't tumor cells, but they're participating in the cancer progression. That can be cells known as, for example, fibroblasts. These are cells that kind of integrate within the tumor and can secrete factors that promote a growth or invasion or migration, or even our own immune system can actually be pro-tumorigenic, so support that tumor progression. We usually think of the immune system as fighting cancer, but in some ways it can be almost manipulated by the tumor to be pro-tumorigenic. And so we really try and understand all these different cell types that are participating in the cancer that aren't actually the cancer cells themselves.
So Nikki, there's, it's so interesting to talk about the tumor microenvironment. Can you tell us, is it the same in all areas of the body, or if you have the tumor microenvironment in the primary tumor and somewhere else in the body in a metastatic site, is it going to behave the same way?
The short answer is no, they do not behave the same way, and they do have different tumor microenvironments, depending on which tissue the metastasis is present in. It's known that as a metastasis establishes in different organs and then continues to grow, it will recruit cells from that local environment that it's in. So if a metastasis is in the lung, there's going to be lung cells contributing, and there's liver cells contributing. So they certainly behave differently because of that. Of interest, when metastasis occurs, it's not always just a single tumor cell that lands in these environments and then, proliferates and makes a new tumor. We know now that there's these circulating clusters that are found in patients, and these clusters contain tumor cells that can be phenotypically different. And it also will take some of the tumor microenvironment from the primary tumor with it as it leaves and spreads and then lands in different organs. So this process is very dynamic, and it's really a contribution of what originally left the primary tumor. And then as it establishes and grows in that secondary environment, the cells that are recruited and then contribute to tumor growth in that secondary environment. One of the things I think about all the time is the fact that, we're pretty confident that even before tumors are detected, there are thousands and thousands, if not tens of thousands or hundreds of thousands of tumor cells that can spread through the body and land in various organs. We're actually very good at it. Our body is able to clear 99.9% of those cells that spread. And in some cases, 100%. Some patients will never develop metastases. So the spread is terrifying, but if anything, it shows how good our body is at defending itself. However, that very small fraction of cells that are able to survive and eventually proliferate and make a new tumor in these distant organs is something that I'm trying to really understand. So, I'm developing tools to be able to track both the factors that those rare cells depend on, as well as communications between those rare tumor cells that survive and the microenvironment around them, trying to really map kind of the critical components that allow those tumor cells to survive and proliferate over time is, I think, an area that would help so many patients. Because if we can stop those rare cells from surviving, then we can certainly help a lot of patients by preventing them from developing metastases later on.
Can you kind of explain to people like what a genetically engineered mouse model is, how you kind of follow cells, that start in one place in the body and end up somewhere else?
Absolutely. This is, what you're getting at is a tool that I regularly use, and that's the, mouse models of pancreatic cancer. These were, developed by some very talented people that closely model the way pancreatic cancer progresses in a human. So it steps through that, early tumorigenic phases that we were referring to earlier, all the way through metastasis. And it utilizes this mouse, tumor-initiating mutations that we know will drive pancreatic cancer in patients, but only in their pancreas. So again, it's this mouse that's developing pancreatic cancer that looks just like human. So it's a very, very powerful tool. And then we can use these, what Susan was referring to as lineage tracing. So the tumor cells will express fluorophores, like let's say they glow green, and that those green glowing cells will then let us track those tumor cells throughout progression. So as they land in the liver or lung, we can then find these very microscopic, early metastatic lesions, or, let them grow out into large metastatic lesions. And again, just track that cell behavior over time. Of note, these mice have been very powerful in testing early, therapies that we believe may improve outcomes, and has allowed us to really understand the tumor microenvironment, again, as we talked about, throughout different phases of tumorigenesis. People that really focus on again, the heterogeneity within the tumor cell population itself, and can we target the very aggressive tumor cells that are driving therapy resistance metastasis, that's happening. There's a lot of good work looking at how the mutations that are causing the cancer can determine, better response to certain treatments over others. So essentially, like, perfecting that initial enrollment into therapy depending on your mutation status. Or, in my case, I try and, again, target factors that are promoting that the ability of tumor cells to survive in distant organs. And so in my lab right now, we're working on factors that are upregulated in the liver of patients and mice that develop metastases in the liver at much higher rates. So essentially trying to help that population at higher risk for liver metastasis.
And you said that liver metastasis is one of the number one places that it happens, right?
Yeah. Susan, correct me if I'm wrong, but the last I saw, about 60% of patients can develop metastases in the liver. So it's certainly a high interest for pancreatic cancer in general.
And I think that just can be maybe even anatomically explained as a very simple way, because all the blood from the pancreas actually has to drain through the liver. And so it's the first kind of site of filtration of cells and the first site where any metastatic, tumor could potentially land. So it's often the most prevalent site of metastasis for most patients.
Exactly. And, there's even evidence that those clusters we were talking about just by chance get lodged in the liver because they are, can be quite large. And so you can imagine just by chance they end up landing there. A lot of maybe listeners today have probably heard of immune checkpoint blockade or immunotherapy. These are kind of really hot areas in cancer treatment. But as it's been hard in pancreatic cancer, a lot of the trials and efforts haven't turned out as promising as we'd hoped. But now there's some evidence that suggests maybe why those didn't turn out so well, and then essentially adding on treatment that may improve those outcomes. So, essentially all hope is not lost for leveraging our own immune system to improve treatment. So I think, we might be seeing some strides here pretty soon in that area. So much amazing research that's been coming out lately, really understanding the different cells in that tumor microenvironment and which of those populations are more pro-tumorigenic versus anti-tumorigenic. I think, in the next 5 to 10 years, there might be a lot of exciting things to come out. It's a little bit of a different journey than others. I grew up farming, but even as a child, I really knew that I was very interested in biology and research. And so my first experience in research was actually in the world of agriculture research. I was working on developing this strain of no-burn Kentucky bluegrass, or even eradicating a fungus that impacts garlic growth. But I learned pretty quickly that I wanted to be closer to human medicine and the clinic. And so when I went to graduate school, I joined a lab that worked on a number of cancers, including leukemia and pancreatic cancer. But I was really strongly drawn to pancreatic cancer. The reason being is, it really feels like this very challenging puzzle. There's been a lot of progress in a lot of tumor treatments, including pancreatic cancer, but there's still a long way to go. So I personally feel like there's a lot that we can contribute to patients that are afflicted with this disease.
All of your work, it sounds really exciting. One of the reasons that we're passionate about this podcast is sharing these kind of new revelations that are coming out, and just to give people hope that, more treatments are coming. And I'm just so thankful for your time today. Thank you for diving so deep into this topic. And it's all really exciting stuff. It's the motivation behind the Sina Magowitz Foundation. All of our funding goes toward research. So it's amazing to hear about the stuff that you guys are doing.
I really appreciate your guys' work in moving pancreatic cancer research forward, and cannot be more thrilled to be working with Dr. Sai and others here at MCW. So
thanks, Miranda, for letting me tag along today.
Yeah, thank you all for your time, and I think that this episode definitely is calling for a part 2. So we'll have to have you back on.
Fabulous. All right.